Hello, General Biology, and welcome to the fifth in the screencast series for Chapter 7, Sections 7.3 and 7.4. And we were just talking about hypertonic and hypotonic and isotonic solutions and conditions that are produced as a result of osmosis. So we're going to start off with this video clip.
Hypertonic solution. A hypertonic solution is simply a solution that has a lower concentration of solutes, dissolved substances, compared to the inside environment of a cell. For example, water is considered a hypotonic solution since it is very low in solutes. A hypertonic solution is a solution that has a higher concentration of solutes compared to the cell's inside environment. For example, water with a lot of salt is considered a hypertonic solution since it is very high in solutes. How can we examine the effects of a hypertonic solution on a cell? You might be thinking, well, I'm made out of 50 to 70 trillion cells. If I drink this, I'll know what happens to cells in a hypertonic solution. But do not do that! Drinking salt water can be very dangerous as it severely dehydrates you. Nausea, vomiting, and even hallucinations are just a few symptoms of drinking massive amounts of salt water. Ask anyone who's been stranded at sea, which is unlikely because people who drink seawater eventually died due to kidney failure. So what you really need is a lab animal to conduct your test. Darn, she's too smart. Time to try her older brother. Well, that was a bad experiment. What's a better way to test how a cell reacts when placed in a hypertonic solution? Some eggs, like chicken eggs, are surrounded by a hard outer membrane which protects the semi-permeable inner membrane. This membrane is semi-permeable because it allows substances to leave and some substances to enter. In particular, the semi-permeable membrane allows water to both enter and leave the cell, but large molecules, like salt or sugar, are too big to pass through. The question is, how can we examine the semi-permeable qualities of this inner membrane with the hard eggshell in the way? Dissolve the outer shell by placing an egg in vinegar. Vinegar has a chemical called acetic acid. Eggshells contain calcium carbonate. When calcium carbonate in the eggshell comes in contact with acetic acid, a chemical reaction occurs. Calcium carbonate will break down to form carbon dioxide, water, and calcium acetate, thus dissolving the calcium carbonate shell. Within seconds, you'll see bubbles form. These bubbles are carbon dioxide that are produced when acetic acid breaks down the eggshell. Within 3 days, you should have a semi-permeable membrane, free of the hard outer shell, to conduct your experiment. Before starting our experiment, we will first mass our egg using a scale. Egg A has a mass of 94 grams. Egg B has a mass of 103 grams. Egg A we will place in a hypotonic solution, water. Egg B we will place in a hypertonic solution, thick and sugary corn syrup. Egg A now has a mass of 97 grams. Egg A increased slightly to 103% of its original weight. We can conclude when a semi-permeable cell is in a hypotonic solution, water will enter the cell. Egg B now has a mass of 59 grams. Egg B decreased to 57% of its original weight. We can definitely conclude that when a cell is in a hypertonic solution, water will leave the cell. In fact, this is evident by the layer of water sitting on top of the corn syrup. However, 103% change in mass is not very significant. So how about we try this experiment again, but switch the eggs? This time, egg A is going in the hypertonic solution, which is still on the right. Egg B is going in the hypotonic solution, which is still on the left. After several hours, mash your eggs again. Egg A went from 97 grams to 55 grams. Egg A has decreased to 57% of its original weight. Egg B went from 59 grams to a whopping 105 grams. Egg B has increased in weight to 177%. From this experiment, we can definitely conclude that when a cell is in a hypotonic solution, like water, water enters the cell. On the other hand, when a cell is in a hypertonic solution, like corn syrup, or more realistically salt water, water will leave the cell. Now that you're done with this experiment, time to play with those shellless eggs.
No, no! You're just gonna...
All right, so I showed you that video because we're actually going to be doing that experiment in lab, and we're going to be doing it with pancake syrup and decalcified eggs, and we're going to see what results you get from every 5-minute interval. You're going to be weighing them and massing the eggs. All right, so what we just talked about were forms of passive transport. The molecules were moving with the flow from an area of high concentration to an area of low concentration. But what if we want to take molecules against the gradient and we want to move them from an area of low concentration to an area of high concentration? For that, we're going to require energy. And this is a form of molecular transport that we call active transport, and it moves the molecules against the concentration gradient. So we're still going to utilize proteins. Here's our phospholipid bilayer, and this is a protein. But in this type of protein that we're going to see here, it's going to be kind of like a gate protein. And in order to open up that gate, we're going to use the energy of ATP. To open the gate up, the molecule will come in, the gate will close off like you see here, and it will pull it in. And notice we're moving from a low concentration to a high concentration with this. So we're going to utilize transport proteins, and the two transport proteins that we're going to utilize are gates, like we just saw, and ion pumps. Okay, here's that word ion. We know that ions happen when they gain or they lose electrons. And when an atom gains or loses electrons, it becomes either positively charged or negatively charged. So in this example that we see here, look, we have sodium ions, positively charged sodium ions, and we have positively charged potassium ions. And this particular type of protein is going to be called an ion pump. You can see it's going to work with energy because of the pumping action that takes place, but it's also going to be negatively charged so that it will attract the positively charged molecules to move across the membrane. So here's positively charged sodium, this is negatively charged, and it's going to move it across. And of course that ATP is going to cause the pump to open and close in a pumping action to help pull these into and out of the cell. So this is a form of active transport. Both require energy. There's another type of transport that is called bulk transport. And again, we're going to have to I think this is a short video, so let me just click on it and see.
The substances taken in by single-celled organisms are often particles or large polar molecules that cannot cross the hydrophobic plasma membrane. Many single-celled eukaryotes employ endocytosis to ingest such food particles. In this process, the plasma membrane extends outward and surrounds the food particle. Cells use three major types of endocytosis: phagocytosis, pinocytosis, and receptor-mediated endocytosis. If the material the cell takes in is particulate, such as a bacterium or a fragment of organic matter, the process is called phagocytosis. If the material the cell takes in is liquid, it is called pinocytosis. Specific molecules, such as low-density lipoproteins, LDL, are often transported into eukaryotic cells through receptor-mediated endocytosis. Molecules to be transported first bind to specific receptors on the plasma membrane. The interior portion of the receptor protein is embedded in the membrane. The protein clathrin coats the inside of the membrane in the area of the pit. When an appropriate collection of molecules gathers in the coated pit, the pit deepens and seals off to form a coated vesicle, which carries the molecules into the cell. Exocytosis is the reverse of endocytosis. This process results in the discharge of material from vesicles at the cell surface to the outside of the cell.
So this part talks about bulk transport, and bulk transport happens in two processes, endocytosis and exocytosis. And this is where we have large molecules or a group of large molecules or waste products that cannot be moved by passive transport or active transport through the proteins. And bulk transport is a form of active transport, but it can't be the active kind that uses the proteins. And it requires bulk transport. It requires changes in the membrane and the formation of vesicles. So endocytosis, endo meaning into, and cytosis is the cell, is a form of taking materials into the cell. So here's a solid food particle that needs to come into the cell, and when it comes in contact with the cell membrane is going to form a little pocket or a pit, and as this pressures a little bit more, this pocket in the pit is going to seal off to form a vesicle, and this vesicle is going to have the food that will come in, and now a lysosome can attach to it to digest it. This form, since this is a solid particle, is known as phagocytosis, and phage means to eat. Pinocytosis is an endocytotic process as well, but this is taking in extracellular fluid. So once again, the fluid molecules are going to form a little pit, and that pit is going to seal off and then bring that piece of fluid or that fluid into the cell. And this is pinocytosis, and pino means small. Now you heard him describe receptor-mediated or receptor-aided endocytosis. And if we have cholesterol or LDLs like he mentioned that are non-polar, and we know that because of the polar heads, water's being pulled in, these aren't going to be able to mix because oil and water don't mix, so they're going to need some help. And along the membrane, there are receptors that are going to be able to hold them. And the receptors will form a pit, and that pit will break off into a vesicle, and that vesicle will then bring those molecules in. So receptor-mediated endocytosis. All right, so here's our definitions. Endocytosis, endo, into, cytosis, the cell, and it's a form of bulk transport that moves molecules into the cell. And there's three different types: phagocytosis, pinocytosis, and receptor-aided. Phage means to eat, so it's going to be taking in large particles. Pino means small, taking in liquid, and endocytosis will be used for those non-polar molecules. We're going to stop here, and we'll talk about exocytosis in the next screencast.